On a more serious note, I just wanted to thank everyone for sticking it out to the end of the conference today. It's a pleasure to be here and to share with you a lot of the work that the students from my lab have done, as well as you'll see me smattering in mentions of some of the folks from Marcelo's lab as well. I just want to announce just a few brief disclosures of companies that I've helped found over the years. But most importantly, I want to thank all the students that contributed to this work. You'll hear me mention So-Young Moon, Madison Kane, especially Ben Yeoman and Pranjali Berry are all former, well, the last two are former graduate students from my lab, as well as all of our collaborators. In particular, some of the physician scientists that we work with, including Ann Wallace here, who's a surgical oncologist and treats all the patients that I'll describe for you today, as well as, of course, Marcelo and all of his great students. We've seen a couple here, Bibiana, Paloma, Mauricio, and so their names will come up as well. And in particular, I also want to thank our funding agencies, in particular in the United States. That would be the National Cancer Institute and the National Institute for Neurological Disorders and Stroke, for what I'll tell you about today, but also then the National Science Foundation. And we're very proud of something called the Wu-Sai Human Performance Alliance, which is I think oftentimes a lot of our funding agencies are really dedicated to helping solve diseases and coming up with therapeutics, but the Human Performance Alliance is actually focused in on athletic performance, and I think that is something that we oftentimes don't focus as much on in science and engineering. I do, just as a plug, want to mention that Marcelo, the check is in the mail. He and I have had a wonderful collaboration, and like I said, I'll mention some of the data from that collaboration, but we literally just recently, as of Monday this week, got a grant. I tried to put the oversized novelty check in my suitcase, but I couldn't, so a PDF of the award notice is about as good as I can do. But the last thing that I want to mention before getting into some of the science that we've done over these years is just that I've been really impressed by the community here in Brazil in cell biology, and would like to welcome you to our community of cell biologists, which conveniently will have a meeting in San Diego in December of this year, and the abstract deadline is next month. So if you are at all inclined to come to sunny San Diego, this is some images of our convention center and then the skyline. That's a QR code that gets you to the website. I would also mention the university in San Diego is a little bit further up the coast, but this is the Sanford Consortium where my lab is located, and then out here is the Pacific Ocean. So we literally are right on the ocean, and it's one of those first world problems, we would call it, where I had to tell people that they couldn't bring their surfboards into the lab. So that was a new one for me when I moved to San Diego, but it's a wonderful community. So, you know, if you're coming to the cell biology meeting, by all means, let me know, and we can arrange for you to come up and have some great fish tacos. This is what we're known for. All right, so without further ado, I'd like to just dive into some of what we work on. And if you're not familiar with this term, mechanobiology, it's really the focus on understanding the mechanical environment that tissues present to cells and how that environment dictates what those cells are going to do, like what their responses are. So this is different from the traditional biomechanics concept where you want to push on an object and feel how soft or stiff it is. So here we're building that environment to challenge the cells to respond in some way. And in doing so, we present the cells with a defined set of extracellular properties. Here you can see a very classic image from a book called Albert's Molecular Biology of the Cell, or Albert is the first author. And in this, you can see lots of stringy fibers. That's our extracellular matrix. The big things that are stuck in the middle of those fibers, those are the cells. And then you can see little tiny nodules that are things like secreted proteins or growth factors on the surface of these cells. So thinking about this sea of spaghetti noodles, or whatever you might want to use as an analogy, those proteins present the cells with a set of defined mechanical properties. And they typically revolve around these four things. I shouldn't say just mechanical, but these four properties. So the first is the composition of that extracellular matrix. So is it mostly collagen, or is it fibronectin, or laminins? And please keep in mind there are 14 different types of laminins. Most people ignore that. The second thing that I would point out is topography. So whether you have a smooth surface or a surface that's rough, we generally would equate this to something like in the basement membrane. So if you have a two-dimensional sheet of extracellular matrix that lines, say, a blood vessel or something like that. Next would be porosity. So how big or small are the pores in your matrix? So here the porosity is actually pretty low. It's a pretty tight set of matrix. And then last but not least, the thing that I want to start out telling you about is stiffness. And so that's the difference between something that's hard, like the podium here, so this would be gigapascal, and something that is unfortunately really soft and has gotten softer over the years, enjoying a lot of beer in San Diego. And so that would be more like kilopascal. So we're dealing with many orders of magnitude difference between what we conventionally will grow cells on, which is tissue culture plastic, versus what they would see in vivo. So it's like 6-7 orders of magnitude softer. So I think if nothing else, I hope that you take home from the first part of my talk an appreciation for the fact that stiffness really does matter. And it matters in a whole variety of contexts. So I'll just say decades ago when I was a graduate student, that included concepts like that stem cells would respond to this matrix and differentiate down different lineages based off of the mechanical properties of that environment. So if it's softer or if it's stiffer, then cells might become bone or fat depending on that microenvironment. So it's not just the growth factors that you provide those cells. But I want to tell you not about stem cells today, but about a story related to cancer and the onset of or tumorigenesis. And conventionally we think about this in a term called epithelial to mesenchymal transition. I've seen a lot of wonderful cancer posters over the past 2 days. So I hope that this isn't review is sort of old hat to a lot of you, but for those that aren't familiar with it, this is a process where epithelial cells will convert and start to express transcription factors that are more related to a mesenchymal phenotype. And what the paper that I'm showing you here is a paper from Val Weaver's lab now about 20 years ago, where she introduced for the first time this concept that stiffness doesn't just matter to things like stem cells, but it also matters to the development of tumors. And I think we can all appreciate that if you're to manually palpate or push on a tumor, that it's physically stiffer. And it's stiffer for a reason, because the tumor-adjacent fibroblasts, or those cancer-associated fibroblasts, are expressing a lot of matrix, and they're also expressing a lot of cross-linking proteins, and that stiffens this microenvironment and provides then a vehicle for those cells to migrate out from the tumor. So what Val did was create an in vitro mimic of this. Now, I think we can all appreciate on the left-hand side that you can take a collagen gel, and you can make a 1 milligram per milliliter gel, and you can make a 3 milligram per milliliter gel. And the 3 milligram per milliliter gel is stiffer. It just physically feels stiffer when you squeeze it. And the cell response is different. So you see that these cells form really nice spherical clusters. We call them acini. But that as you increase the stiffness from left to right, you see a change in the cell's behavior. And the reason for this, we think, is mechanical in nature, that the cells lose their polarity and undergo EMT. Now, the problem with a system like this is that two parameters inherently are coupled together. The first would be mechanics, but the second is adhesion. Because you're making you're presenting more matrix to these cells. There are more sites for them to adhere, so they can stick to more stuff. And so you're sort of conflating two different parameters in this system. So if we then look on the right-hand side, this is a classic polyacrylamide gel system. So if you've ever run a Western blot and you've resolved your proteins out on a gel, typically you're going to use SDS page, or the PA part is polyacrylamide, right? So that's what your gel is made out of. Now what Val did was she took this system and sort of tilted it on its side, attached a matrix protein onto the surface of this gel, and then added her cells on top. Now, that's a two-dimensional system. These are 3 dimensions. So how did she accomplish this? Well, she added some matrigel on top, and that enabled these cells then to form these spheroids. And hopefully you can appreciate in the softer microenvironment, and the absolute numbers here are not as important as just knowing soft from stiff. So for mammary tissue, it's really, really soft, so 150 pascals. That's sort of the consistency of Jell-O. I don't know if that's a thing. Okay, yeah, Jell-O. Versus something that's really rigid, 5,000 pascal, which, you know, think of pushing into your muscle, let's say. So you can see dramatically how the behavior of these cells changes. Now, over the years, we've asked a variety of questions related to what are the intracellular regulators of this process, how you convert something that is an inherently physical signal into a biochemical response. And so some of the papers are highlighted there in red that we've been working on. But I'd rather not talk about that today because those are all published efforts. And I'd rather share with you some newer work that we think is really exciting. And that are 2 questions in particular. So the first is what happens when the cells form these 3-dimensional structures before stiffening? And then what happens when these cells migrate from the stiff tumor into the soft stroma? So those are the 2 questions that we're going to ask. So the first one needs a little bit of material science, so I'm just going to walk you through it. If you've never seen an NMR plot, that's what we're looking in panel A. And it basically just shows, hey, we modified the backbone of our material. This is a hyaluronic acid, so we're methacrylating it. And what that allows us to do is to initially cross-link our environment, our matrix, and make it soft. We can do whatever we want with the cells there. And then later, we can cross-link it again, and we can make it stiffer. So now you have a material or an environment that goes from soft to stiff. So that's the basis of what we're going to do, and we're going to ask 2 questions in this context. So how does acinar development change with different stiffnesses? And then how mature do these acini need to be to see if they can become less stiffness sensitive? So what we first did was we said, okay, well, let's plate these cells into a soft gel. So that's for a week. And then we came back and said, okay, now let's stiffen them, and we'll stiffen them for another 5 days or 10 days. Now in the soft context, where we're essentially not changing anything, they form these big hollow acinar structures. Great. But now if we start to stiffen them a little bit, we don't see a response. We stiffen them, an order of magnitude. You see a little bit of a response, and then all of a sudden, boom, once it's really stiff. Now I should also mention that in the static environment, what Val's lab found was that somewhere around, say, 500 or so Pascal, so roughly 3 times stiffer than normal, that's when they began to see cells undergoing EMT. Well, so if we look at the same data set, but now we're using a dynamic system, we actually see that most of our cells are still remaining in this spheroid condition up to, 1,000, maybe at least 1,000, where you're getting about 50% response, and then there are 30% non-responders at 3,000 Pascal. But this is a graded process. So we were really intrigued by this. Now, the second question that we wanted to ask was not necessarily a gradient in stiffness, but we wanted to ask a gradient in time. So here, the same sort of experiment is done, but everyone is plated on the same day, and then we chase by the number of days that we allow the cells to remain in a soft environment and then convert it to a stiff one. And what I want to draw your attention to is the quantification here, where you can see that by day 10, so this is 10 days in a soft environment, and then you stiffen it to 5,000 Pascal, you can see that there is a small but non-zero number of acini that remain intact, which was really surprising to us and suggests that there's some collective signaling, if you will, between these cells. Now I just want to share one final slide from this work, and the idea was that for the signals that are involved in this, so the key hallmarks here would be TGF-beta signaling, would be an EMT marker like twist, and so we're looking at the cytoplasmic to nuclear ratios of these two, or I should say nuclear to cytoplasmic. Now in a stiff or a soft only microenvironment, one that doesn't change ever, we see very nice separation of these two populations. So if it's soft, then nothing is nuclear localized, there's no signal. If it's stiff, now all of a sudden there's a lot of signaling, and everything localizes to the nucleus. In the case of a dynamic environment, we looked at both cells that were responding and spreading out and undergoing EMT, and cells that weren't responding and remained acinar. And hopefully you can appreciate in panel D here that there's just no correlation whatsoever. And I'm skipping through a lot of data, but the take-home message from this signaling was that there are multiple redundant pathways that cells use in these dynamic environments that they don't use in a static environment. So in the static system, you knock out one thing, it just goes kaput. But in a dynamic system, there's a lot more signaling between cells and crosstalk that allows them to use these multiple pathways together. So if we tried to knock out YAP signaling or SMAD or twist, we needed an inhibitor for all of them to see the cells then fail to respond to stiffening. So I want to just briefly switch gears and focus on that second question that I mentioned at the outset. And that is, okay, well, so now once these cells have undergone EMT, they've begun to spread out, they need to disseminate and leave the tumor and invade the stroma. So how do they remember, or do they even remember, what happened in the tumor, right? Do they have any sort of memory of what happened? Because as they're disseminating, they're leaving all their friends and they're going out on some journey where they're going to be surrounded by stroma that's not familiar with the tumor. So this is a series of images from patients from Marcelo's institute. Excuse me. And I hope this cartoon really nicely illustrates sort of the transition from normal mucosa to a dysplastic lesion and ultimately to a full-blown squamous cell carcinoma. Now, the detail that we're trying to layer into this is the concept that this normal mucosal layer or tissue is relatively soft. And as the tumor develops, it gets stiffer. And once those cells leave the environment, how do they deal with that? And so this is a concept that we call mechanical memory. Now, one of the great things about our collaboration is if you go back 10 years and you search the literature for stiffness and breast cancer, you find thousands and thousands of papers. But if you do the same search for oral squamous cell carcinoma, you get 0. Well, you did before our collaboration, I should say. So now you get a couple. And I think that there's a growing community that recognizes that this concept of the mechanical environment being really important is not just specific to a certain subset of solid tumors, but is a widely applicable concept. So one of the first things that we did, or I should say that Bibiana did, when she came into my lab on one of these exchanges, was to look at a series of different both epithelial and mesenchymal cell lines derived from oral tissues. And of course, you get the predictive behavior. Right? Epithelial cells like to sit on the top, mesenchymal cells like to invade. But we wanted to put that concept together with our engineered environments and say, okay, well, if we now make the environment stiff, what happens? And the Cal27 cells, that's the epithelial line, all of a sudden they start upregulating a lot of EMT markers. Now the SCC25, that's a mesenchymal line, they're not upregulating things because they're already expressing a lot of them. But so it's an acquisition of this phenotype as they're undergoing EMT, and that's why you see the Cal-27 cells responding. Now, going to the patients from Marcelo's clinic, one of the things that Bibiana was able to find was the fact that if we graded the patients based off of the tumor stage that they had when they presented in the clinic, and then you go back and you check how organized this collagen is. And 7-8 years ago, the way that we identified softer or stiffer tumors was by how organized their collagen is. We now do atomic force microscopy on it, but that's another story. And what you can see, though, is that higher stage patients, so stage 3 and 4, there are more patients that had organized tumors. And if you look at this Kaplan-Meier plot, you see that they actually do worse. So, of course, stage 3 and 4 patients have worse outcomes. But if you were to look at the coloration here, the patients that have organized collagen do worse regardless of stage. So whether we're talking about stage 3 and 4 or 1 and 2. So there's this stratification, if you will, based off of mechanics. So if we fast forward a few years, So Young, one of my graduate students, took up this idea and was working with Paloma in Marcelo's lab. And they got this idea that they could grow these cells in a soft or a stiff environment and then switch them and challenge them and see, do they quote unquote remember the behavior that they acquired in a prior environment. And so we can do this either by replating the cells, or now we actually do it by dynamically cross-linking the environment, so we can make the gel softer or stiffer over time. But I'll walk you through this one set at a time. So purple to purple, that's just soft forever. So the cells stay epithelial. If it's orange to orange, then they're stiff forever, meaning that they undergo EMT, and now all of a sudden they're really invasive. They like to crawl everywhere. If we look at our mesenchymal cell line, that's a control on the right-hand side. That's stiff forever. Of course, they like to invade. We've already established that. But now if we look at this condition that we call learn, and that's orange to purple, that means that it went from a stiff microenvironment for about a week or 5 days, and then we transition them to a softer environment, whether we could degrade the material or we trypsinize them and replate them. Regardless of how you do it, these cells are in a soft environment, yet they're looking much more like they're invasive cells, which to us was really perplexing, and it's a term that we like to call mechanical memory. Now, we can do this chemically as well, so we can use LPA, lysophosphatidic acid, and that will enhance the contractility of these cells and sort of biochemically make them develop mechanical memory. And then we can use other drugs like blebbistatin to knock out this property. But we wanted to delve into the signaling that was being used in this system, and so we ran a phosphokinase array, and the thing that was really striking was that as these cells were learning, they're expressing a lot of phospho-AKT, and when they've disseminated, now all of a sudden they're expressing a lot of phosphofocal adhesion kinase. So one of the things that we did was, again, to go back to Marcelo's patients, and we looked at these stage 3 and 4 patients, and we said, okay, well, how much phospho-AKT do they express in their tumor core versus the invasive zone? And what was interesting was that we saw this correlation again between the cells in the tumor core expressing a lot more phospho-AKT and then shutting that down as they left. So we thought that this could be a signal for the cells to learn in their stiff environment, and then once they're in that softer environment, they don't need to learn anymore, so they don't really need any more phospho-AKT. Now, this whole signature, we looked at the transcriptome of these cells and developed a sort of signature of what learn looks like, and then what don't learn or forget looks like. And what I mean by don't learn or forget is just that we block the cell's ability to sense this environment. So that's where they don't learn. And if you do a comparison of their transcriptomes, you see about 800 genes that are differentially expressed. A lot of them are related, not surprisingly, to the matrix, cytoskeleton, adhesion, and migration. And when you plug all of this into the Cancer Genome Atlas to sort of in silico predict how patients respond, you see very clear differentiation between patients that do well, relatively speaking, in this don't learn or forget category versus the patients in the learn category. Now, why does this happen? What we think, and hopefully that's what we will be studying over the next 5 years, is the idea that this memory, all these pathways that are getting activated are activated because they adopt a specific epigenetic signature. So in collaboration with one of Marcelo's clinical colleagues, what they were able to show is that in fact patients that do better, clinically speaking, tend to have higher H3K9 acetylation. So Mauricio, when he was came up to my lab from Porto Alegre, one of the things that we started honing in on were the transcriptional signatures that we could find. So there was a lot of methyltransferases and expression of histones in the learn condition versus, say, DNA repair proteins in the, that are downregulated in the same condition. So that hinted that maybe something's going on, but what really struck me was the one result that Mauricio had, which was when you grow these epithelial cells in a soft microenvironment, they acetylate their histones more. So this epithelial signature really wants to be acetylated, just like the tumors that survive better. So we're doing a lot of follow-up experiments as part of that grant, but I would just like to close by saying, hopefully thus far, what I've been able to convince you of is the fact that the extracellular matrix is really important, but not only its properties when you start your culture in vitro, but how those properties evolve over time. And it's really this time evolution that's important for your cells, whether they're undergoing EMT in a dish or they're trying to migrate out into a collagen gel or something like that. All of these properties are really important. So in the last, say, 15 minutes or so, I'd like to switch gears. We're part of a very large consortia in the United States with the National Cancer Institute, something that we call the Physical Sciences and Oncology Centers. And what the PSOC has done over the last 10 years is really shine a spotlight on the contributions that physicists and engineers like myself, in terms of our original training, can have on this field. And so we've looked at a variety of different mechanical properties that cells have to figure out, is there anything that's sort of determinative? And I'm going to focus in on adhesion as being our sort of property of the day. And the reason why we're so, so interested in identifying what these properties are in these cells and how we can better characterize them is that if you look here, this is just a summary, it's a few years old now, but it's a summary of all the different biomarkers that people have published in literature over the last couple of decades, trying to isolate biological signatures of cells that tend to be more metastatic. And the idea was, well, if we can find that one holy grail, that protein that's indicative of metastatic disease, independent of the cancer type, wouldn't that be great? And unfortunately, we haven't really found that yet. But if we go back to the concepts here of sort of physical properties and how that might dictate the responses of these cells, if we think about a solid tumor, well, every solid tumor, the cells have to detach. They have to then disseminate and migrate through their adjacent stroma. They have to undergo transendothelial migration to get out into the bloodstream, and then ultimately do the opposite process once they find the site of secondary disease where they want to form that tumor. So thinking along those lines that maybe physical properties are a universal marker, we got started. And so this is about 10 years ago. Our first generation device was to take cells, put them on a cover slip, put them into a very large vat of fluid, so this holds about 1 liter of buffer, and then spin them really, really fast. like 5,000 RPM. So this thing, this motor, when it's running in the lab, you literally can't talk because it's that loud. But the point is that what we're trying to do is apply shear stress to these cells. So shear stress, the concept is if you have a surface and you have fluid moving over that surface, that fluid causes some friction. That's why when you drive an internal combustion engine, you have to have motor oil, otherwise those parts will rub up against each other and they'll stop, right? So that's shear stress. So we want to do the same thing, but we want to cause shear stress for our cells because that will cause some of them to detach. Now I promise you, I only have 2 equations in my talk. My engineering colleagues would kill me for this, but I only have 2 to spare you. But this equation here equates the amount of shear stress, or tau, to a lot of very important properties. And the property that's most important is R. And that just means the radius away from the center. So if you're in the center of this assay and you spin around, you don't really feel any shear stress because there's no fluid moving about. But if I'm at some distance R away from the center, now all of a sudden, as I can't walk around the circle that I want, but as I'm moving around, I now experience some shear stress because the fluid above me is I'm moving and the fluid above me is staying still, but I'm rotating through some distance. So that causes a shear stress, and in panel D you can see what the cells do. They detach. And that's illustrated here in the insets. So you get, once the cells are spun, you get a bright spot in the center where all the cells are attached, and the cells at the edge, unfortunately, detach. Now what we were able to do with this is to look at a series of different cancer cell lines, breast cancer cell lines, and see what behaviors they had. So you can look at MCF10A. That's a non-malignant, non-metastatic breast cancer cell line, epithelial. And there's, they're relatively well adhered. If we do the same experiment for MCF7, this is a non-metastatic but malignant cell line. Again, you don't see any change. Now I want to point out that red versus black indicates cells that either do not have divalent cations or cells that do, or I should say the buffer does, right? So these the MCF10A and MCF7 cells don't really change a whole lot, relatively speaking, when you remove divalent cations. But the MDA MB231, this is a triple negative, very aggressive cell line. You put it into mice, they make beautiful tumors. They are very well adhered when you have these divalent cations around, but they're not, this red data, you can see a huge shift, 2.5 orders of magnitude difference when you withdraw the divalent cations. Why does this happen? Well, magnesium, manganese, and calcium are really important for integrin activation. So in the presence of those divalent cations, you get really robust adhesion for all these cell lines. But if you withdraw it, the one cell line that changes is the metastatic one. Why is that? Well, as it turns out, the microenvironment immediately adjacent to the tumor is very, very poor in magnesium, manganese, and calcium. So this is a mechanism that these cells have evolved that helps them migrate out from the center of the tumor. Interestingly enough, we've confirmed this across breast cancer, lung cancer, prostate. We've even recently, with Frank Fernari at UCSD, also shown that this works in glioblastoma. So even cancers that are only locally invasive undergo the same sort of process. But again, why are they doing this? It's because of these divalent cations that are preserving the focal adhesion structure. So in the presence of these ions, or in the absence in the MCF10As, they're relatively insensitive. Their focal adhesions stay well assembled. In panel H here, you can see in the 231 cells, that is not the case. The moment that you withdraw these divalent cations, all of a sudden, their focal adhesions start screaming and they disassemble. And they become very weakly adherent. Now, the problem with all these assays is that whether you're in the center or you're at the edge, you're seeing different forces. So there's no way when you detach for us to know, did you detach with a lot of force or little force. So enter our engineering concepts. Second equation, my apologies, this will be the last one. So this is an equation that relates the amount of shear stress to the geometrical properties of our device. And this device is a flow chamber that allows us then to apply the same amount of force or shear stress to all of these cells. So hopefully everyone can appreciate that even in the United States, our first generation 0 assay, we literally use binder clips to hold the thing together. You know, figure out is there a there before you really do fancier engineering things like this is a microfluidic setup. This is a higher throughput setup where we have 3 channels. We use this when we're doing mouse experiments because you can sort like 10 million cells per chamber. But we also like to make smaller versions that sit on our microscope. And I will show you this version. So here, there's a lot of engineering that goes into this, but it's a diverging chamber, meaning that the forces change depending on your position. But I'll go over that later. The big take-home message is that when you sort these cells into weakly adherent or strongly adherent, they do different things. So the weakly adherent cells in our 231 breast cancer cell line, they travel a long distance, they move very quickly. The same story is true with lung cancer cells. So these are 1299 lung cancer cells. And the reason why these weakly adherent cells are so effective at doing things like making tumors, as I'll show you in a second, is because they are insensitive to the same sorts of gradients that we see in tumors. So the tumor is stiff, the surrounding is soft, right? Most cells undergo a migration process called durotaxis, where they migrate from soft to stiff, except for cancer. These cells migrate from the stiff tumor to the soft stroma. And what is exemplified in our data is that you can make regions of your gel that's soft and regions that are stiff. And the cells, when you plate them at time 0, are evenly distributed. But at, say, 24 hours later, now you can see an accumulation of the strongly adherent cells, that blue SA data set, and you don't see the same accumulation with our weakly adherent cells. Same story is true with PC3 prostate cancer cells and H1299 lung cancer cells. You're seeing an increase in this, what we call antidurotactic or adurotactic behavior, which just means that the weakly adherent cells are actually able to migrate down these gradients. So that's one nice thing. I will just as a quick plug, we just filed some US patent paperwork to make these durotactic assays in high throughput. So if you're at all interested, before our company is started and the university shuts me down, and we have to sell off the IP, I'm more than happy to make these and we can ship them anywhere in the world. But you've got 96 little assays where you can do this same experiment. Okay, so apologize for the digression. I just want to mention one more thing, and that is that we can again sort these populations, we can sequence them, and we see about 500 genes that are differentially regulated. And if you plug in those differentially regulated genes, they all happen to again relate back to migration and to the cytoskeleton, surprisingly not to adhesion, but that's probably because it's more of an assembly process than an expression. And again, whether we're looking at progression-free interval or disease-free interval, we see very nice separation of our 2 patient populations. Whoops. And I should mention that that is for triple-negative breast cancer, but we've also done it for invasive ductal carcinoma and invasive lobular carcinoma. We see the same separations. And all that's fine and good, but I want to conclude with 2 final thoughts. And the first is we need to show that adhesion really matters in a mouse model. So we can take our cells, we can sort them into weakly adherent or strongly adherent, and then our unsorted population as well. We see no differences in the primary tumors as they're developing, but we see major differences in the metastases that they make. So 231s love to go to lung. You can see a huge upregulation in the weakly adherent population, whether we're using normalized or unnormalized data. Normalized is in the middle, because all this is done in a pairwise manner. We can also not just do this with 231 cells, but with their companion cell line 468s, and they do the same thing. So we are really pleased with that. Unfortunately, all we're doing is taking these cells and injecting them into a fat pad. that doesn't really mimic what a patient sees. So our clinical collaborator, Ann Wallace, yelled at us and said, "That's a terrible animal model. Make a better one." And so we went back to the drawing board and we said, well, what does a patient see? Well, they see a tumor that gets resected after, in this case for the animal, a few weeks. So what we did was we injected these cells, or we sorted the cells, injected them into the mice, and then after 3 weeks, we resected the tumors, the primary tumors. So those are gone. We leave the fat pad. You can see at 3 weeks, the lungs are basically negative, so there's no GFP signal. But then after 12 weeks total, we come back and we look at the lungs. And I was really shocked to see that weakly adherent cells just light up. Or the weakly adherent, the lungs from mice that received weakly adherent cells light up, meaning that the cells that had migrated out into the tumor adjacent stroma, into that fat pad, over the first 3 weeks, caused all these secondary metastases. Since the lungs were clear at 3 weeks. So we thought that was really interesting, and finally believed us and said, well, maybe there's something there. So one final thing that we could do with these sorts of assays is that we could intentionally mix different cell populations together to try to mimic what was happening in the mice. And you can see in very nice sort of stepwise fashion in our co-culture system, you see sort of one behavior in purple in this first arc, and then you see a second arc. That is indicative of the two different cell populations that we've mixed together, 10As and 231s. What that allows us to do is to create a prediction. So this heat map in panel C is a heat map that we constructed. It's basically a risk score, whether that mouse was more or less likely to develop secondary metastases. So on the x-axis here, it's how adherent the cells are, again, that tau. Right? And so the more weakly adherent, the further to the left they are on this graph, the more likely they are to see secondary mets. Also, just the number of cells that came out, so the cancer fraction, we refer to that as the number of cells that have disseminated from the tumor, and if you have a lot of those, that's also bad. So it creates this ring, if you will, on the top edge of this heat map, and so we were really impressed by that. And in fact, if you were to do something called a receiver operator curve, which is device speak for how accurate is your device, what's important is something called the area under the curve, which for us is 0.94, and that's really good. But the problem with mouse tumors is that they look nothing like human tumors, mouse tumors that are made with human cells. Whether it's an injection or whether it's a patient-derived xenograft, they're just terrible. So Ann came back and said, hey, let's do an investigator-initiated clinical trial. So we did that. One of the complications of doing patient samples and trying to isolate cells is that there are a variety of different cell populations that are present, not just our epithelial cells that have undergone EMT, but you've got all those fibroblasts that are mixed in, you have adipose. Mouse fat pads aren't actually very fatty relative to human mammary tissue, so it was a huge problem. We ended up having to do some sorts, but ultimately the take-home message is that we actually do see different signatures, whether we're talking about patients that are in our control population in what we call reduction. So this is a reduction mammoplasty. It's a woman who's coming into the clinic to have some breast tissue mass removed for no reason, for reasons that are beyond just having a tumor present. So this is a purely quality of life issue. So they're disease-free versus patients that are coming in for a tumor and they're getting that resected. And you can see in panel G here that we actually get halfway decent separation in our patient population, only having looked at about 15 or so patients thus far in our clinical trial. But what's more important, what I'm more impressed by with Madison and her data, is that if we were to look at that cancer group, the 10 or so patients that we've looked at thus far, we see really nice stratification between patients that come in the clinic and by histology are diagnosed with ductal carcinoma in situ versus patients that are diagnosed as invasive, whether it's ductal or lobular carcinoma. Now, why is this separation really important? Well, it turns out about 15 to 20% of all patients that come in the clinic with breast cancer are misdiagnosed or sort of undertreated in the end. So those patients are the ones that typically will reoccur within a few years. Our disease will reoccur. So we hope that an assay like this is a better or an alternative means of stratifying our patient populations, and that will enable us to more accurately inform a patient of what might be the appropriate standard of care for them. Now, we're doing this all with resected tumors, right? So they're already well into their disease progression. They've also been treated with a lot of drugs to that point. So some of the complications here, we've had patients that have had really weird data because they've been on things like Ozempic, or they've been on a lot of different triple ACT treatment or neoadjuvant, and that has complicated our study. So one of the things that we want to do after completing that trial is to move into core needle biopsies. So this is sort of pre-diagnosis when the patient has first come in, say they've had a positive hit on a mammogram, and we want to better stratify the patient at that point rather than just relying on histology. So our next goal is to do the same sort of study with core needle biopsies. So that way we can stratify patients and say, well, if you have more weakly adherent cells, then maybe you're in this green curve here, and the likelihood of disease is a lot higher, so maybe we should be a bit more aggressive in our treatment. So hopefully we can say some of those more sort of prognostic statements in the years to come. But if nothing else, if I could just close by saying, hopefully I've convinced you that the way that tumor cells adhere to their environment really depends on the environment itself, and that the percentage of those cells that are in fact weakly adherent might be an interesting biophysical marker that maybe at some point in the future will have some prognostic value to it. So with that, again, I'd just like to close. Thank you for your attention, for all of your hospitality. It's really been fun to be here over the last couple of days. I really appreciate all of your science, and hopefully I'll see you in San Diego in December. Thanks.